Hi everyone, in this video we will discuss few questions from acid-base disorders. So the first question is pH. Here you should write the definition of pH and the nature of solution if there is any change in pH and what is the clinical significance of this pH measurement. So by definition, pH is the negative logarithm of H plus ion concentration. So if the solution pH is 7, then the solution is said to be neutral, and if the pH is less than 7, then the solution is acidic in nature, and if the pH is more than 7, then the solution is basic in nature. So what is the clinical significance of this pH measurement? This pH measurement helps to diagnose the acid-base disorders, that is acidosis and alkalosis. Then you should write what is the normal pH of this arterial blood. The normal pH of arterial blood is ranging from 7.35 to 7.45. Here the lower limit is 7.35 and the upper limit is 7.45. If the arterial blood pH is less than 7.35, then the condition is known as acidosis. If the arterial blood pH is more than 7.45, then the condition is known as alkalosis. So these are the two acid-base disorders. One is acidosis and another one is alkalosis. So in acidosis, the arterial blood pH will be less than 7.35, and in alkalosis, the arterial blood pH will be more than 7.45. And the second question is regarding the respiratory acidosis and respiratory alkalosis. It's a 3-mark question. So here you should write the arterial blood pH depends on the two factors which determine the arterial blood pH. One is the concentration of the carbon dioxide, second one is the concentration of the bicarbonate ion. And the concentration of carbon dioxide is regulated by respiratory system, whereas the concentration of bicarbonate is regulated by renal system, that is kidney. So concentration of the carbon dioxide and bicarbonate regulate the normal arterial blood pH. If there is any change in this carbon dioxide concentration or bicarbonate concentration, that will produce changes in arterial blood pH. Then you should try the normal arterial concentration of the carbon dioxide. The normal arterial blood concentration of carbon dioxide is around 40 millimeter mercury. So what is respiratory acidosis? Acidosis means the arterial blood pH should be less than 7.35. So when acidosis associated with high plasma concentration of carbon dioxide, that is carbon dioxide concentration should be more than 40 millimeter mercury. 40 millimeter mercury is the normal carbon dioxide concentration in arterial blood. So when this arterial blood carbon dioxide concentration is more than 40 millimeter mercury, when it is associated with acidosis, then that condition is known as respiratory acidosis. Then just write this heading, that is the biochemical findings seen in respiratory acidosis. What should be the biochemical changes? Just write these 3 points. One is pH, partial pressure of carbon dioxide, and bicarbonate concentration. So in acidosis, in any type of acidosis, the pH should be less than 7.35. Here the change in pH is due to the change in carbon dioxide concentration. So the carbon dioxide concentration here it is more than 40 millimeter mercury. Here the bicarbonate concentration is normal. That's why it is known as respiratory acidosis. And the second one is respiratory alkalosis. Alkalosis means the pH should be more than 7.35. Sorry, here it is 7.45. The arterial blood pH should be more than 7.45. When this alkalosis associated with low plasma concentration of carbon dioxide, then it is known as respiratory alkalosis. So what are the biochemical findings in respiratory alkalosis? The pH should be more than 7.45. Then it is known as alkalosis. So for alkalosis, the pH should be more than 7.45. Now what should be the carbon dioxide concentration? It should be less than 40 millimeter mercury. The normal carbon dioxide concentration is 40 millimeter mercury. Here it should be less than 40 millimeter mercury, and the bicarbonate concentration is normal here. And it is known as respiratory alkalosis. And the next question is regarding the metabolic acidosis. And what are the causes for metabolic acidosis, and what is metabolic alkalosis? So in this type of question, just write the concentration of bicarbonate also determine the pH of the arterial blood, and the bicarbonate concentration is mainly regulated by the renal system, that is the kidney. So whenever there is a change in bicarbonate concentration, this will produce some changes, their corresponding changes in the arterial blood pH. That type of acid-base disorders due to the changes in bicarbonate concentration is known as metabolic acidosis or metabolic alkalosis. Then you should write the normal arterial bicarbonate concentration, that is 24 milliequivalent per liter. So what is metabolic acidosis? So in any type of acidosis, the arterial blood pH should be less than 7.35. Here bicarbonate represents the alkali, whereas carbon dioxide represents acid. So here the acidosis due to low plasma concentration of bicarbonate ion, then it is known as metabolic acidosis. So what are the biochemical findings seen in metabolic acidosis? The pH should be less than 7.35 and bicarbonate concentration should be less than 24 milliequivalent per liter. 24 milliequivalent per liter is the normal bicarbonate concentration. Here the carbon dioxide concentration is normal. Then that condition is known as metabolic acidosis. What are the important causes for metabolic acidosis? So endogenous production of acid. For example, diabetic ketoacidosis and ketoacidosis in starvation, lactic acidosis in violent exercise, high-grade fever. All this produces metabolic acidosis. And the next question is regarding plasma buffers. Here, just write the definition of buffers. Buffers means the solution which can resist the changes in blood pH when an acid or alkali is added. So what are the different types of buffers which are present in our body fluid? These are the three different types of buffer. One is bicarbonate buffer, second one is phosphate buffer, and the third one is protein buffer. Then just write few points related to each buffer. Bicarbonate buffer, it is the major buffer in the extracellular fluid. In case of phosphate buffer, that is the major intracellular buffer. And in case of protein buffer, the buffering activity of this protein is due to the presence of histidine. Histidine is an important amino acid. So those protein having maximum amount of histidine residue, thus protein having buffering activity. Examples are, one is albumin and hemoglobin. This albumin and hemoglobin having high histidine residue. And the last question, that is anion gap. So by definition, anion gap means it is the difference between measured cation and anion. Here the cations are sodium and potassium. These are the major cations of the blood, and major anions are the chloride and bicarbonate. So the difference between this measured cation and anion, that is known as anion gap, and the normal range is around 12 plus or minus 5 millimole per liter. So you should write what are the conditions which is associated with high anion gap. And high anion gap is associated with some acidosis, that is some ketosis state like starvation ketoacidosis, diabetic ketoacidosis, lactic acidosis, toxic ingestion like methanol poisoning, excess salicylate intake. So all these conditions are associated with high anion gap acidosis. So that's all regarding acid-base disorders. Thank you.